Proofreading new DNA. So once you make the new DNA, there's a certain amount of mistakes that are made, which are called mutations, and your body has enzymes that will go back and fix them. So DNA polymerase, that's the enzyme that puts the nucleotides in to making the DNA polymer. Anyway, this enzyme initially makes about 1 in 10,000 base pairing errors. So a mistake in DNA is called a mutation, and mutations can be bad. They can make a mistake in the DNA that can cause a problem in, let's say, one of your skin cells or liver cells that's reproducing. Or if it happens in an egg or a sperm, then it could be a very bad mutation that could affect the baby. On the other hand, once in a while, mutations are really good and they're helpful to the population or to the individual, and then it's spread through the population. And sometimes mutations are neutral and they don't really matter one way or another. But any kind of change in DNA mistake is called a mutation. Only mutations in germ cells can affect the next generation. So these are the cells that become egg or sperm. And enzymes proofread and correct these mistakes. So we'll talk about those a little bit. This is actually really important. I want to go back to this just for a second. If you have a mutation in your skin cells, it can it can give you cancer, for example, but it's not going to affect your babies. But if you have a mutation in an egg or a sperm, then that's what's going to affect the offspring. So you can put a star around this, or maybe a few, because that's actually pretty important stuff. DNA damage and repair. Chemicals and ultraviolet light radiation can damage DNA in our body cells. Cells must continuously repair damaged DNA. So excision repair, that means when you go in to excise something means to cut it. So you go in and you cut it and fix it. That occurs when any of over 50 repair enzymes remove damaged parts of DNA and fix them. DNA polymerase and ligase, there are several different types of DNA polymerase, but one of them, anyway, will go in with DNA ligase and replace and bond the new nucleotides together. So let's do some quiz time. The sides of the DNA run in opposite directions, and so you can see this is 5' to 3', whereas this one's 3' to 5'. That's called anti-parallel. And it's movie time, and I think we're done. Some of these videos are actually really fantastic, but I don't think they're going to show on this.